Okay, we see the chromosome. Okay, so another explanation here. So related to chromosome. So before it replicate, okay, the chromosome will have only one chromatic. Okay, before it replicate means before it start to divide. So when it's ready, it will change from chromatin to chromatic. Okay, chromatin to chromatic. So from chromatic, it will replicate itself. Okay, replicates into sister chromatic. Okay, sister chromatic. So that means one is chromatic. Okay, when it replicate, it become sister chromatic. So before it want to divide, it's called the chromatin, which we cannot see. Okay, it's invisible. Invisible. Chromatin invisible. Chromatic is visible. Okay. So after replication, they will have two sister chromatic, and this sister chromatic are hold together. Okay, by the centromere. Okay, the centromere will hold it together. Okay, this is the centromere. Okay, the centromere will hold the sister chromatic together. Okay, now during mitosis, the two chromatic of each will separate. So this chromatic, right, they have duplicate themselves. Okay, they duplicate themselves. So one will go to one daughter cell. Okay, so the duplicate copy will one separate at the centromere into half. Okay, so one go here, one go right. Okay, one left, one right. So that means we will produce two daughter cell. This is how the mitosis happen. Okay, so mitosis will produce two daughter cell. And then you remember that the diploid cell, they have two copies of each chromosome. Okay, where we call it the homologous chromosome. So the homologous chromosome will come from the father, paternal, mother is maternal. Okay, so but this homologous chromosome, they are not attached together. So we will see what is homologous chromosome. Okay, later on, after this type. Okay, now first thing is the terms. So terms that we can use in cell division, most of the time we will see. Okay, the diploid, haploid. Okay, actually we have started with the words. Okay, so what is diploid? Diploid means the number is 2n. 2n means even number. So even number. So for example, human, you have 46. Okay, the housewife just now you have 8. Okay, so that is certain. You no need to remember the number of chromosome number. Okay, chromosome number no need to memorize. Just for human, you have to know only. Okay, so that means from 46 chromosome, if I put into pairs, they have 23 pairs of chromosome. Okay, human have 23 pairs of chromosome, and then the diploid cell will have one copy of each chromosome, one from each parent. So that means each of these chromosome, one is from 23 pairs. Yeah, that means 46. Sorry, 46 means 23 from father, another 23 from mother. So later on we see meiosis, you will understand that. Okay, so haploid is when the chromosome number is half. So we use the word n. 2n divided by 2 will become 1n. Okay, so it will have 46 divided by 2. That means from the homologous chromosome 23. Yeah, so 23 pairs. Cut in the pair into 2. Okay, so that you will have 23 one daughter cell. Okay, 23 chromosome, another 23. We cannot use pair anymore for haploid. Okay, no more pairs. So what is homologous? Yes, this is homologous. So we can see here in the pictures here. So this is one chromosome. This is two chromosome. Okay, so there are two chromosomes, same length. Okay, same length, same size. So this is called the homologous. Okay, sister chromatic is here. This is sister chromatic where after they duplicate. Okay, this is homologous chromosome. So they will carry the same type of gene but different characteristics. Different characteristics mean if this one carry the gene for the eyes, this one will also carry the gene for the eyes. But this one will carry the gene for blue eyes. This one carry the gene for brown eyes. So whether you have brown or blue, it depends on dominancy. Okay, who are more dominant, the blue eyes or the brown eyes that we have? Okay, if the blue eye is more dominant, that means you have blue eyes. This chromosome will be shown. This one will be kept. Okay, that's why we will see more in genetic later on, the next the chapter behind. Okay, so now sister chromatic is another term that you have known. Okay, sister chromatic is after duplicate. Okay, after duplicate. Now this is the karyotypes that we can Yeah, we can see here karyotype. So when I mentioned just now karyotype, maybe you are not clear, so now you see the word. Okay, karyotype means if I take out the individual chromosome and I put it in I extract it, I take out means that I extract it from the nucleus and I rearrange and I count. Okay, I will have, if it's a human, I will have 46. So if you need to count, you can count like this. Okay, this is 1 2 3 4 5. Here we have 23 pairs. Okay, right, this is 24. Oh no, this one is all. Okay, so you have 23 pairs. Okay, this is plus xx or xy, so 23 or you can see the word or there. Okay, so that means 1 2 3, you can count. This is one pair, sorry, 1 2 3 4 5 6 7 8, and then you can see 23. Okay, so one pair, two pair, three pair. So what I want you to see here, this is chromatin. Okay, chromatin means, you see, one pair is one chromatin, two chromatin. Okay, there are two chromosomes here. Okay, two chromatin. As for when they're ready for different to divide, okay, to divide, one here, one pair receive got 1 2 3 4 4 can say chromatin, but actually there are two chromatin where they each of them duplicate. So you see the X one. Okay, so it's still 23 pairs. Okay, 1 2, they have number actually down there, no need to count. Okay, if you get the slide, you can just see the number. Okay, so 22, the last one. Okay, 23, the 23 pair. Okay, the 23rd pair is actually XY or YY, XX. Okay, XX or XY. This one is the sex chromosome. This is the sex chromosome where they are different a bit. Okay, if they are XX means they are woman. Okay, XY means they are men. Okay, so this is how we differentiate the chromosome of a man and a woman. Okay, so this is karyotype. Maybe too much info for you, but you have to do a bit of revision and you can repeat the videos again and again. Okay, so these are extra notes that you might need to know. So you can just see, I think. Where is the thing? Yeah, it's here, but it's not projected or it's not shown when I make it in slide form. Okay, so it's in a notes form. Okay, so you can see down here 19 20 21, these are extra notes for you. So we straight go to the cell cycle and the mitosis. Okay, cell cycle and mitosis. So first thing, of course, you have cell cycle. Say cell cycle, just now when we say cell dividing, okay, the cell have to undergo, before it can divide, it have to undergo a series of preparation stage. Okay, preparation for divide. Okay, for example, you want to go vacation or travel, you have to prepare yourself, you have to pack your bag and so on. So well, for the cell, the same thing. So every cell, in order to divide, they have to undergo this cell cycle. Okay, the cell cycle means you will go one round starting from G1. Okay, it get ready and then divide. After that, finish dividing. So finish dividing, the daughter cell might have to divide again. So it repeat G1 as G2. So they produce another daughter cell. After that, it repeat again. That's why it goes circle and then a circle. Okay, when it goes circle circle, we call it the cell cycle. Okay, so some cells, so we have different type of cells, right? So beside the normal cell, the gamete cell, among the normal cell, we have also have the different type of cell. For example, the nerve cell or the skin cell. Okay, so we will see that parts of cell. Okay, not the sex cell or the gametes. We are seeing the normal cell. From the normal cell, we divide them. They have different type of cells again. Okay, so they may be the skin cells. Alright, so for the skin cell, the gut lining cell, they will divide rarely or never. They will divide constantly. Okay, constantly means all the time they are dividing. So for embryo, it means the baby, the skin cell, the gut lining cell. So they will divide all the time. But other cell, they will divide rarely or never. Okay, For example, the nerve cell. Okay, the nerve cell, they're not really dividing all the time unless you have injured your nerve cell. Okay, like accidents or what. Okay, then it will replace or repair. Okay, so when they divide, okay, they must go through a circle of event. So this is what we say the cell cycle. Cell cycle is the circle of event, and then in order to get daughter cell, okay, to get daughter cell. So the most of the time, okay, the first cell cycle, the main cell cycles are the interphase. Okay, so the interphase are these three stages: G1, S, and G2. This is called the interphase. And then the last one, the second phase, is the M phase, the mitotic phase. So G1, S, and G2, they are the interphase. And then you have mitotic phase. Okay, so cell cycle consists of two major phases, which is the interphase and the mitotic phase. So if we see here the another cycle, this one is more complicated one. So it starts from G1 here. So it starts from G1, okay, then S, then G2. The pink one is still, the orange pink is the interphase. Then after that the M phase, see, M phase or the mitotic phase, it will contain mitosis and cytokinesis. So the last one C, see, C, and then the blue one, purple, C, cytokinesis, and the purple one and the blue one will be the M phase. Okay, this is the one cycle for the cell. Okay, so mitosis and meiosis are single steps in the cell. So when we say mitosis and meiosis, means the nucleus division. The nucleus division is actually one step, meaning to say start from G1, S, G2. If to produce a normal cell, okay, autosome cell or the somatic cells, okay, to produce somatic cells, they will undergo mitosis and then stop, then two daughter cell produce. Okay, but if it's a meiosis, after G2 it will undergo meiosis and then stop, finish. Okay, then G1 and then G2. That one is for meiosis. Okay, understand? They are all single step in the cell cycle. Okay, single step means they go one round, then they stop. Then another daughter cell, if they need to divide, then they will start with G1 again. Okay, if the same parent cell need to divide again, then they will start with G1 again. That means they go one round and then they stop. Okay, that's single step. So cell not in the process of dividing are in the, if they are not divide or they are in prepare of divide, that means most of the time the cell are in interphase. Okay, so the chromosome are duplicate and prepared for the next division, okay, in the interphase stage. Okay, so they are preparing all the time. So different type of cell will have different rate of cell division. So some cell, the developed cell. Okay, this is the repeat for just now. Okay, in more detail. Some cell they are not going to divide. Okay, never divide. Okay, never divide. For example, nerve cell, skeletal, red blood cell, and so on. Red blood cell they never divide. They will just after 3 months they will be destroyed at the liver. Okay, at the spleen. Okay, so this one red blood cell it will not be divide. Okay, they will need to produce again. Okay, some cells are resting state until they are signal to do division. Okay, some cells are like that. Okay, they will wait until there's a signal for them to divide, they divide. But some cell will continually divide. Continually divide means all the time is going to divide. Okay, now there is a gut cell, the lining of the wall. Okay, now we see interphase. So what is interphase? So interphase, you will see is a time or is a stage where the cell or we can, to be more exact, where the nucleus prepare themselves to divide. Okay, so in the interphase we have G1. So G1, the cell will grow rapidly in size. So we have high metabolic rate in here during this time, and then high protein and cellular organelle is synthesized. So a lot of organelles are being synthesized. They use a lot of energy. After that they enter the synthesis. Synthesis is where the DNA is replicating. So this is the place or this is the time where you can see the sister chromatic is formed. And then you have the last one, which is the G2. So G2, they will start more growth occur. Okay, they will start this fiber starting to form. So you can see here, okay, here the small picture here, spindle fiber means come from the centriole. Okay, remember centriole that you have learned in chapter 2. Centriole involved in the division, cell division. Okay, so this centriole will move to opposite. It will start to go to opposite. It will start to form the spindle fiber. Okay, later we see what is spindle fiber, and it is actually generally you can say interphase is the phase where they get ready. Okay, they get ready. So then we will see the mitotic phase. Okay, mitosis. Okay, what is mitosis? Okay, so the cell will divide to make more. The chromosome must precisely divide. So that means they during interphase also they must make sure all the chromosome already divide correctly. When they divide and duplicate, they must photostate it correctly, exactly the same. Okay, so that it will not have anything miss out or anything too much or anything double or anything extreme. Okay, add on. Okay, so mitosis is for the normal cell. The normal cell here we have another term, we call it somatic cells, and it will go through the life in all parts of the body. So in all the parts of the body, meiosis is only for the gamete. So somatic cells will divide by mitosis. So one cycle involved of mitosis. When we say mitosis, it's only one cycle. One cycle means from one mitosis only lah, and then it produce two daughter cell and from one parent cell. And then the parent and the two daughter cell will be genetically identical. This one you have to memorize. This is the word that we use for exactly the same. Okay, exactly the same in biology, we will use the word genetically identical. So the parent cells are diploid, and so the daughter cell is also diploid during mitosis. So this is what happened. One parent cell, after mitosis, it will get two daughter cell exactly the same, genetically identical. So what we can see, we have M phase, okay, anaphase. After interphase we enter M phase, the mitotic phase, and this mitotic phase are in 4 stages. So prophase, metaphase, anaphase, and telophase. You cannot miss or you cannot just jump out. It must go from prophase to metaphase, anaphase, then telophase. Okay, telophase. So what is prophase? So prophase is when the chromosome condense. You see, now it become like extinct. After interphase it become the extinct, and then the nuclear membrane starting to break down. Okay, so see, alright, the centriole move to opposite, spindle fiber starting to form. You see, so nuclear membrane, okay, nuclear membrane disintegrate, so the chromosome is going out. This is plasma membrane. This is nucleus membrane. The purple one is nucleus membrane. So after that, this one you just read up. This is the writing of it. After that, after the nucleus membrane disintegrate, okay, all the chromosome will align in one line. Okay, it will align in one line. We call it metaphase plates. Okay, metaphase plate. That means after prophase, everything go one line. The spindle fiber, the centriole will reach the opposite pole, and then the spindle fiber, which is the yellow thing. The yellow thing is fiber. Fiber is microtubule, made of microtubule. It will join to the centromere. Okay, we will see here what is centromere. Yeah, this one you just read. So I need that centromere. Where is it? Yeah. Okay, so this one. So this is the blue green one. Okay, this is the one that we call the spindle fiber will attach here, and this yellow part or the centromere. Okay, the centromere will have this kinetochore where it is attached to the spindle fiber. So when the spindle fiber contract, the blue thing contract, it becomes short, and then this thing will become halved. Okay, it will break into two. So during anaphase, this is the one I want to say. So spindle fiber contract, so it becomes short, so it will pull the sister chromatic to opposite side. Pull it to opposite side. Okay, so that is anaphase. You can just read. Anaphase is when it pull it to opposite side. Okay, last stage, which is telophase. So telophase, it will start to make cleavage furrow, and during this time the nucleus membrane started to form again. The reformation of the nucleus membrane is formed again. Okay, you have one centriole here, one centriole here, and all the organelle will be in two. Okay, so last stage. Okay, so this telophase is when they form the